The next step, because the goal here is we want to sequence the exact A's, T's, G's, and C's in our barcode gene. To do that, first we need to make lots of copies of just the barcode gene. So we're going to use a process called the polymerase chain reaction, or PCR. What it does is it copies DNA. From one copy, you can get billions. So I'm going to draw the shortest, easiest, simplified schematic what PCR is, and we'll send you a video that does a better job. But let's pretend this is a piece of my fish DNA. And my barcode gene is just part of it. So this is my 16S ribosome DNA, aka my barcode gene. And this is only a short region of the DNA in the fish. So there are millions of base pairs in this fish genome, and I want to copy just this region. So with PCR, what we do, if we're going to build DNA, we need to tell this reaction where to start building. And where to start building, we specify with short pieces of DNA called primers. So we have one that faces forward, one that faces backwards. We call them forward and reverse. And we're going to add an enzyme called, so these are our primers. So we need, so we need primers. We need an enzyme to build DNA. So that enzyme is called DNA polymerase. Enzymes are named by what their job is. So if something ends in ACE, A-S-E, that's telling you that this molecule is an enzyme. It's a protein with a job. Polymerase means, so a polymer is joining things together. So this is a DNA joining together or building enzyme, okay? And then we're also going to need the building blocks to make DNA, or DNTPs. So these are your A's, T's, G's, and C's. And you can just buy this stuff, okay? So what's going to happen is we're going to mix our fish DNA with these primers, the DNA building enzyme, and the DNA building blocks, and we're going to run it through a machine that changes temperatures to build the new DNA. So again, we'll send you a video that tells you exactly what it is, but essentially the too long didn't read, mixed all together, come back in an hour and a half, you will get just the barcode gene times billions of copies. So that's the too long didn't read. You put the stuff in a tube, it does its job. Its job is to build DNA. It will build you billions of copies, right? So I'm going to set it up as an example. Hopefully it'll work well, and we'll include it in your analysis. So what I've got is I need to set up 3 reactions. So I have my 2 fish samples, fish 1 and fish 2. And then just to make sure I didn't cross-contaminate things, I'm going to set up a sample where instead of adding DNA, so this is my fish 1 DNA, hopefully I will get billions of copies of just the barcode gene from fish 1. This is DNA from fish number 2. And this third tube is water. So if I made a mistake and accidentally splashed DNA everywhere, this would show if this would create DNA as well. So this is just what we call a negative control. We are hoping that it doesn't make me any DNA. Okay, so this is just part of the standard what you include. All right, so what's going in the tubes? Well, I have my fish DNA. I'm going to add what's called a a PCR mix. So it's okay, cool. You just buy it. This is really pretty color. So this tube has our DNA polymerase enzyme. It has our building blocks, and it has the salts and buffers to let the enzyme do its job. It's also pink because it has a loading dye already mixed in it for the next step, which is to run your samples out on agarose gel. So you just buy it. It's pre-mixed. So great. They didn't have this when I was an undergrad. I had to mix all this stuff together on my own. So this is the DNA building enzyme, the DNA building blocks. And now the enzyme needs to know where to do its job. Which part of the DNA does it need to build? Well, these are, again, you buy it from a company. These are my primers. Tubes look empty, I promise they're not. They have very tiny volumes of a very concentrated piece of DNA that is perfectly matched to the forward part of my barcode gene and the reverse part of my barcode gene. So these will tell that enzyme where to start building. Then if I add the fish DNA, we're good to go. So you do have to do a little tiny bit of math. So I want my total volume to be 25 microliters. This mix comes at double concentration you want. So I want half my volume to be the mix. I already did the math. I know I need half a microliter of each of these primers. And I want maybe 5 of DNA.
So actually,
How much you want?
2x is wrong.
My choice is wrong? Oh gosh, it should be, yeah, half of 25 is 12 and a half. Thank you, Mother Dr. Burnett. It's been a long time since I've done this. All right, so 13 and a half plus 5 is 18 and a half. I want it to be up to 25, so that would be 7 and a half water in each tube. Well, I don't want to have to pipette 4 different things into each of these tiny tubes, plus the DNA. So that'd be 1, 2, 3, 4, 5. I don't want to do 15 little pipette steps. Did I do it wrong? What did I do wrong?
12.5 plus 1 plus 7.5 is 21.
So 6.5.
Yep.
Ah. See, this is why it's good to have backup. Probably would have worked, but not as well. Okay, 6.5. All right, so what I'm going to do is I'm going to mix all this stuff together and portion it out, and then I can customize each tube adding just the DNA. So I'm going to make enough for, let's do 3 and a half. Okay, so 4. Oh, jeez. Okay, I have permission to do extras.
We always do 1 extra.
Okay, 1 extra. All right, so that would be 2. 6.5 times 4 is should be 27.
26.
26.
6.5 times 4 is 26.
All right, 26. Hey, I am not rocking the math this morning. Not gonna lie. 12.5 times 4.
50.
Huh?
50.
Oh, 50. Okay. You sure it's not 48? 50. Okay.
48 plus 2.
Plus 2. Right.
Yep.
Okay. Cool. So that's my math. So I'm gonna mix all of this together.
Is 80. 80 divided by 4 is 20. You're right.
Okay. So I'm gonna mix all that stuff together into 2. Okay. So. You know, as they say, the more math you take, the worse you are at basic math. I went past calculus, so this is why I apparently can't do basic multiplication anymore. All right, so first I'm going to add my DNA, my tech, the pink, very pretty. Okay, so I'm going to do a check mark that I did. That's how I keep track. All right, first primer, I need 2 microliters. Actually, and for that, I'm gonna pull out my even tiniest of tiny micropipettes. This only goes up to 2 microliters. Now I'm gonna add my primer, so my polymerase knows where to start building. So this is 4 primer. Got 2 microliters, it's really tiny. That's that right? Yeah, that's 2, right? Yeah, okay, yeah. And 2 microliters of my reverse primer. Now the polymerase knows where to start building. Okay. 26 of water. I don't see any water on my bench.
Maybe it should be.
Oh, hey, all right, tiny tube. I was looking at the wrong thing. I was looking for one of these bigger ones. All right, so water, like so. Okay. And I know you probably don't believe me, but this isn't enough DNA master mix to make billions of copies. It is a tiny volume, and this is enough for 4 pretty good sized reactions. So I'm gonna flick it, nice and mixed. All right, now for my tubes. Ooh, all right, I got a couple colors here. You can tell me which one you want. So we like to keep things a little pretty. So I've got your choice, pink, blue, or green. Anybody have a preference? I heard blue?
Blue.
Blue, okay. We'll do blue. Blue it is. Ooh. All right. So these are what we call microtubes. They are very small. And I'm only gonna need 3 of these, so I'm going to carefully remove 3 and save the rest. They're kind of spendy. I don't see any scissors. So I'm just gonna do old school and twist. All right, save the rest. Okay, now these are really small, but I still have to write numbers on them. So I'm gonna label them simply 1, 2, and 3. And to be really sure, I'm going to label the lids too. Who knew that having tiny handwriting was an important skill for science? If you give me a superfine Sharpie, I can write 6 letters on these things in each tube. But don't need that today. So what I'm going to do now is I'm going to add 20 microliters of this water, primers, and MyTech into each of those tubes. New tip every time, just to be safe. If I did it right, there should be some left. Good. That means that I measured carefully. Now I'm going to add 5 microliters to each of my templates, and I'll show you what this looks like. It's a very small volume compared to what we're used to in everyday life. So. Alrighty, so let me show you how tiny this is. All right, I'm ready to copy DNA, and that is 25 microliters, of which we only need 1/5 of it, or 5 microliters, to send to the sequencing core to learn the exact A's, T's, G's, and C's. That means that we have another 20 microliters that we can use for other purposes. So one thing we're going to use is what's called gel electrophoresis, and that's a way of a quality control where we can check that first, did our DNA amplify at all? And if so, does it look like we made a lot? And does it look like it's the right size? So we should know ahead of time how big this barcode gene is based on our primers. So we like to do a quality control by running it out on agarose gel before we send it off to the core. So I'm going to go put this in the thermocycler machine to start it heating. And the other Dr. Burnett can tell you about the gel electrophoresis for a little bit.
It would be good to get a thermal cycler.
Yeah, let's put it in the regular camera, right?
Hey, but we can't take the count.
Oh, we can't?
No.
Oh, damn, okay. Yeah, okay, never mind, he's going to bring us a thermocycler, and it'll be awesome.
So you can have to take it anyway.
Alright. You know what? And I need to give, so now DNA's in there. Good to go. This, my handy dandy husband has brought the thermocycler. So, I don't know if you noticed in my previous drawing, when I was drawing like the thermocycler and the heat block, or the heat block and the centrifuge and all that, I was drawing everything as a box. It's not just because my art skills aren't fantastic. It's also because pretty much everything in science looks like a box anyway. So you may as well draw a box to represent it and then just write down the details of what it does. So a box with heat waves coming up, that's a heat block. A box with snowflakes above it, that's an ice bucket. A box with a spinning arrow, that's a centrifuge. Box that says PCR on it, that's a PCR thermocycler. So, yeah.
Let me see.
Is the program stored in here?
You'll have to find it. I don't think we're gonna be able to see the.
No, it's got a pretty big glare on it.
Yep.
Yeah. No, can't see it at all. There's a
Maybe when you.
right over it.
Yep.
Oh, poo.
Well, the best we can do.
I can't zoom.
It is what it is. Alright, so saved. So we've done this reaction a couple times. I'm gonna see if I can find the protocol in here saved already so I don't have to type it in. Fish barcoding, there we go. Okay, that looks right. 52 degrees anneal?
Sure.
Alright. So this machine is just a very precise way of controlling temperature. This enzyme works best with certain temperature parameters. When you learn about PCR in detail, you know you'll learn there are 3 steps. Denaturing step, where the DNA is a double-stranded molecule. Those double strands need to come apart. So this heats up to a high temperature, 98 degrees. That'll melt them apart. Then the primers have to bind to the barcoding gene. That process is called annealing. So you have to lower the temperature down, the primers anneal, and then the DNA polymerase enzyme works best at 72. So we heat it back up to 72. And that continues 40 to, I think we do 40 repeats. So denature, anneal, extend, and that cycles. That's why it's called a polymerase chain reaction. And that will create billions and billions of copies of our fish barcoding gene in each of those tubes. So with any luck, and this would take about 1 hour and 20 minutes, it'll be done.